I'm Suveksha Bastuna, a researcher and public engagement manager at Research Institute for Bioscience and Biotechnology, RIBB. I would like to express my gratitude to all our participants and guests for joining us today for our 4th episode of Bio-Medicum seminar series. As a housekeeping rule, I would like to request you all to mute your microphone during the presentation. The presentation will run for 30 minutes, and 15 minutes after the presentation is allocated for the question and answer session. Today, I would like to welcome and introduce our speaker for the webinar, Dr. Sailendra Pradhananga. Dr. Sailendra is currently involved at region Skane Center for Molecular Diagnostics at Lund as a bioinformatician. He has been working to develop large-scale cancer genomics, whole genome panel and sequencing platform in clinical setting. He is also looking over the communication and presentation of variant results to clinicians and variant curators, and also looking over the implementation of production codes in large high-performance clusters. Following his doctoral degree from Sailendra Laboratory, KTH School of Engineering Sciences in Chemistry, Biotechnology, and Health Sciences for Life Laboratory in Sweden, Dr. Pradhananga has also worked as senior genetic data analyst in Deerwalk Genomics at Deerwalk Services in Kathmandu, Nepal, and as a bioinformatician software engineer at TigerQ in Lund, Sweden. We are very pleased to have him here today, and now I would like to request Dr. Pradhananga to present his talk.
Okay, thank you very much for the warm introduction, and I look forward to talking with you. Okay. I think you guys can see my screen, or how's it going? Can you see my screen, or?
No, we cannot see your screen.
Okay, let me try it out again. Do you guys? Now probably you can.
Yeah.
Yep.
Yes.
So good afternoon. It's around 12 o'clock. It's a lunchtime for me, so my energy could be down. But in any ways, I'm looking forward for the good session. But yeah, I had my talk is titled as Precision Medicine at a Diagnostic Lab. Yeah. Okay, something happened. So basically, I want to first introduce myself. As I can see, I'm Sailendra Pradhananga. I'm a bioinformatician at Center of Molecular Diagnostics, and clearly my research interest is cancer and complex genomics and application of the bioinformatics pipelines in this complex and cancer disease genomics. And also, I'm like most nowadays, I'm more interested into AI-based assisted decision making in different kind of diagnostic project as well. Beyond that, here are my Git links and stuff like that. And I like to run more and do some talk about Arsenal and football in my NOS research interest. But that's not where we are here for. Of course, we are here for the presentation. So in general, what I want to do is I want to get into more detail or get some background on precision medicine, because the world is moving towards it, and how precision medicine is leading to big data bioinformatics and enabling precision medicine. Then what we do in the lab, how we do it, and probably we can end our talk in some kind of a data lab consideration about a data lab establishment in Nepal or something like that in general. Yeah, that is what my general outline is about. So further ado, I will start with first just give an idea about how the things are working at this current scenario. So in this current scenario, I think what I feel like is that, or at this moment, what we're doing is like we are looking for one blockbuster therapy and using that therapy, for example, to treat all of the patients, irrespective of their genetic diversity or differences in their lifestyles and stuff like that. And what we see from that is that the drug, some of them will have a positive effect in the patients, while some of them will not have no effect, and some of them could have an adverse effect on top of that. So on top of so because of this kind of since drug that we use and disease diagnostic is not taking into account the patient stratification, we see a different kind of unwanted effects being going on as such. So basically now there is also been a lot of hype about the or people have been using it of that about precision medicine, which is based upon like use of individualized therapy. When we are back in my like universities or bachelors, it was more hype as a personal medicine. Now the term has been changed to more into precision medicine and stuff like that. The idea is saying that the idea is like we took into consideration patient variability. It's much more proactive approach where you look into different kind of analysis method and you stratify the patients because based upon these different kind of assays, let it be like blood assays or DNA-based diagnostic or other different assays to stratify patient to high risk, low risk, or no risk group, and then give a therapy based upon that. So as to look into account of all of this variability of that. So it's termed as more as a proactive approach in medicine. Has been hyped out by the early paper that published by Hood or something like that. I forget the name at the moment. But most of them is at this current day and age is been driven up by the large scale data and different omics technology out there. But then we have to look into consideration that how is precision medicine a big biology? And when you consider the cells at the moment, it's more, there is a lot of things happening out there. But because of the different kind of high throughput experiments, it's also called as like next generation sequencing based approaches, you're able to integrate different parts of the cells or genes or genomes. For example, you could integrate genomes with the sequencing technology, epigenome with different kind of chip assays or ataxic assays, and you can also look into different RNA expression levels so as to integrate different kind of expression transcriptome in a cell. And on top of that, you can add different kind of proteins. So what I'm basically trying to say that precision medicine is interrogating each and every part of the cell so as to generate out multi-omics approaches, which have generated a high volume data at high velocity. And clearly there has a high value in the clinical setting. It is now thought that by 2025, all of this genomic data, I've been loosely using the term genomic data in order to define all of these approaches that I've been talking about. It has sequencing or astronomical data is subjected or future to be generated out. So in consideration with the precision medicine, there are different kind of moving parts. You could see that there is a lot of research interest or different kind of assays that we've been talking about. On top of that, there is different kind of medical data or clinical utility data, ranging from electronic health records, different kind of images. On top of that, everything is been powered by huge and big computational infrastructures, let it be like biometric data, bioinformatics structures, or different kind of machine learning or other techniques in order to help out, in order to get out more patient-specific multi-omics data, which can be used for the patients in general. So clearly, the idea out here is like there is a lot of patient-centric data has been generated. A lot of patient-centric data is generated out with all these omics approaches in order to provide the right kind of medicine for the patients in general. Yeah, there has been a lot of hype and a lot of, at least in Europe, what I see is that there is a national level initiative for different kind of in order to integrate this kind of approaches or genomics-based approaches in different parts. Most clearly, are famous of them is NHS England approaches, where they have used different kind of they have developed biobanks, and from that biobanks have used have collected a lot of huge cohorts of patients, and then have developed this Genomics England concept or project. Well, of course, I know more about other one, but in Sweden itself, we have very own national initiative. We call it Genomic Medicine Sweden, where we are trying to integrate all of those large-scale omics-based approaches in our public healthcare system, which is being facilitated by different kind of genomic medicine centers all over Sweden. And clearly, our area of focus is more geared towards rare diseases, cancers, infectious diseases, and different other complex diseases. Of course, I work in Center for Molecular Diagnostics. It's a part of Region Skane. Region Skane is a government entity out here, so it's a part of the clinical genetics, pathology, and molecular diagnostic. It's also a part of a Genomic Medicine Sweden, where it's one of the genomics center at the situated in the southern Sweden. And we do a lot of NGS-based diagnostic of various diseases that I showed already. But the interesting aspect is that we are a bunch of clinical geneticists, bioinformaticians with asterisks, lab technicians, and biomedical analysts. Bioinformatician with asterisks in a way that we do more than just bioinformatic analysis at the moment. So that's with an asterisk out there. Yeah, the key area and disease area of focus is more cancer. Like for the cancers, we do different kind of childhood cancers, diagnostics, hematological diagnostic with whole genome sequencing, or transcriptome sequencing, and different kind of gene panels. Clearly, for childhood cancer, one of the examples is neuroblastoma and sarcoma. And for we have different kind of panels and different kind of assays, which has been associated with that. Clearly, we were also doing different kinds of SARS-CoV kind of diagnostic or animal tests during the pandemic in 2020. And recently, I went through our data, and we were able to send or supply more than 14,000 plus NGS tests in 2021. So that's quite a huge volume of tests and diagnostic we have been sending out to the clinicians out there. But then I work in a bioinformatics team. As I said earlier, it's an asterisk because we not only do the bioinformatic analysis, we are working with different kind of lab management tools, development and maintaining of the infrastructures for large-scale data analysis, and of course, developing different kind of bioinformatic analysis pipeline, improvement of it, routine application, and also development and commission of the new analysis. So in general, we are not subjected to only bioinformatic analysis, but we do more than that. Of course, I want to get some, since we are interested in how the data flow, I wanted to give us general overview of how the computational architecture works out there. First of all, the doctors comes to us. We have the lab server, which is based upon LIMS. LIMS is a laboratory information management system, which controls where the patients get registered. And then based upon the samples we get, we send it out from the sequencing machines, which is done as the sequencing is completed. We convert the raw data into FASTQ files. That is the files that is from that we do different kind of pre-aligned QC, and then we supply it our database. But at the same time, we send that FASTQ files to our computational servers, where we run different kind of bioinformatic pipelines to generate the post-QC files and all of the BAM files. BAM files are the align files, the result files. VCF files are also the result files, where you would, if it is a cancer patient, where we call variants in the samples. And then all of these result files are then feed into our database. And then from that database, we get into our front-end services, which is then interpreted by both doctors and clinicians and gone back to the clinicians in a way. So in general, this is how basic computational architecture is works at CMD, where we are working with a different kind of high-end computationals as well as a different kind of servers, and we are tracking patients as well as aligning patient, as well as we're providing different kind of result files to the doctors. I wanted to get more detail into this these workflows. So if you talk about laboratory management systems, we have different sequencing machines. And what we do in the laboratory management system as a bioinformatician or as a system admin is we try to automate the protocols. We run all the sequencing runs, develop the QC, quality control parameters for that. And then from these sequencing machines, we transfer raw data to our servers for ad management. Most of these sequencing machines that I talked earlier, the Illumina machines and the Nanopore as IM S5, they all provide this as their commercial laboratory information system, where we, this is from the environment, that is from the Thermo Fisher, and base space is clearly from Illumina, where we log in our credentials, and then they provide their boilerplate information system. But then what we do is we kind of add more add-on analysis and generate different kind of workflows on top of that. So we kind of host this kind of a LIMS system in our server itself. And it is also responsible to talk with our local server and also talk with our sequencing machines. On top of that, the local servers that is hosted in our hospital or in our in-hospital, as in our centers, we have 2 local servers, which is quite big, and it is basically the main area where all our bioinformatician and IT guys work. On top of that, we have different. It has different data. Different incoming data and outgoing data are managed in this server. It also communicates with our computing clusters, and more importantly, it is only server where we have internet access on it. As I told you, most of it is responsible for incoming sample data management. What I mean by that is different kind of metadata information about samples, about the analysis that is performed, about the diagnostics that has been supplied to us by the clinicians. And also we provide all the wave services. All our reports are generated by this wave services. It is also manages our lab and instrument databases. Also, we provide different kind of interpretation tools from these databases, and it's also managed locally within the hospitals since it's very critical. It's very sensitive as well, and yeah, we do have internet access, but it is very, very restricted. On top of that, we have a computing clusters. What I mean by computing clusters is these are big machines where we run large-scale analysis. Of course, there is not one, but then 16 of those, 16 nodes of 192 GB RAM of 2 into 14 cores, 2 times the 14 cores. It's wrong out here. It should be 15 cores, 2 into time 15 cores out there. And yeah, these computing clusters are the workhorses. They do all kind of calculations, all kind of heavy lifting, data analysis. They run all of our pipelines. This is our production pipeline in general. And yeah, this is, since we run different kind of patient samples, it's very, very restrictive, and it doesn't have any internet access to it. So it only talks with our local cluster. So it can only talk with our local cluster and communicates with the our computing clusters. So basically, I've been talking about infrastructures. Now I want to go into more NGS pipeline developers. So what are the essential skill set? So, or what are the things I use into my day-to-day operations? So basically, I use Unix or Linux operating system, and basically with the bash scripting on that, on top of that, and programming, of course, Perl and Python. These are my stuff. This essential skill set I'll talk to more about in the coming up slides as well, but then I'll talk on a few of them because the rest of them, if I won't go deeper into it, it will be too long and beyond the scope of the presentation. So I'll only talk a few of them. So Unix, I mean, of course, Windows is the most common operating system out there, but we do not use Windows because, it is too messy for us. So we kind of avoid it, and most of the bioinformatic pipelines are not geared toward Windows. It's more geared toward Linux and Unix operating system. It gives the command line interface and helps, and you could do a lot of scripting stuff in there. It's everybody in the NGS analysis or in the bioinformatic analysis use Linux, and most important stuff or most cool stuff is that you can chain together simpler stuff like opening of files or closing of files or extracting information from hundreds of files. This can be done with simple command line interface, and you can make a hell lot of things with a Unix script and stuff like that. So that's why we have a lot of NGS pipelines are developed based upon Unix, are based in Unix programming languages. These are all the pipettes. If you are with the wet lab, pipettes is one of the most used stuff in labbing processes. So yeah, Perl, the programming language is our pipette. In our infrastructures, we are using a lot of Perl and Python. Perl because it's mostly legacy code. It has been going on, it has been coming up from the earlier generations, and a lot of our scripting is done by the older generation. So Perl is more legacy, but Python is where we do all of it's our workhorse, or what I use most. It's used for not only for the analysis script, also used for the glue script to change to different kind of in order to glue or communicate between our different servers, and to pull out data from or to communicate within different servers. Beside that, Python is also powerful. It can do a lot of more backend stuff. And basically, we have been using Python-based front end, called as Flask. in order for our web services as well. So that is what we have been using in our languages. I want to give some idea about or impart some knowledge on Conda as well. Conda is this package manager. That means it has a huge list of softwares where you can directly download it and easily manage an environment, because there might be cases where when you develop your stuff in one computer, it might not work with the other computer because of the different kind of discrepancies. So in order to manage that, we use different kind of Conda environments so that the computers and the softwares that works within my machines can also work in different geographical locations as well. It's one of the open software. You guys can use it as well for your analysis. It's very cool. Yeah, basically what I wanted to get some idea about bioinformatic pipelines. So if you consider like the one of the pipeline that has been working around, the FASTQ files, that is generated by sequencing machine, it goes through plethora of different softwares. So each of the workflow, each of the boxes out here is a analysis pipeline and different kind of a mechanism that is going on. So clearly you could see that the data that goes from FASTQ files, it goes to different analysis softwares and pipelines, methods in order to come to final stage of a PDF and final stage of reporting in our services. So clearly this is like a chain going on. And if you don't know what thing goes on there, it can be very messy. So you would like to have some kind of workflow so as that it is easier to manage stuff, or what things are going where. So sometimes it's very easy when you can do those stuff. So for those stuff to make workflows and through pipeline, there is clearly two main players that is going on in the that we are using. SnakeMake is based on Python. Nextflow is one of the most used ones, and it is getting quite a hype these days. It uses Groovy, which is very similar to, which is a extension of a Java, but then it's very, very simple. But if you learn, but if you are into more Python, we learn SnakeMake as well. So it would be interesting if you are interested in that. You can tell me as well. But yeah, here I wanted to give you an example of how we host our pipeline. So one of the examples is this. This is my common pipeline, or the one of the pipeline that I use, which should show it, but probably it won't show. Let me try to pull it out. So clearly, this is the one of the pipeline that I have been developing around. It's a Nextflow generation pipeline, and here you can find all of the stuff that is required for this pipeline to work. So clearly, what we do is like we develop all of the pipelines and we store as a repo, as open source, so that everybody else can use as well. So this is my common pipeline, which I use, but then there is a community level effort as well, which is called NFCore. And the interesting thing about NFCore is a community level pipeline. It's an open sources. You can easily either want to contribute, or if you are just a user, you can just use it as well. So I would suggest you to, if you're looking or if you're interested in just bioinformatic pipeline, NFCore could be one of the starting point. And quite recently they have been setting up this introductory level classes as well. There are free courses. We can check it out in the website, NFCore pipelines. Yeah, you can. Yeah, I just wanted to give up how does the pipelines, or what are the pipelines that is being used for NGS data analysis. Finally, after going through the pipelines. We also have a reporting tools. All of the stuff that we generate out from the pipelines, we have to send back to the clinicians, but then they would like to see it in some kind of wave interface. So this is one of the wave interface. Here, based upon different diseases, we have different open source as well as in-house developed interpretation tools. One of the examples on which I've been currently working on is cancer as well, Q&A, we call it Q&A, where we report different kind of SNPs, different kind of CNVs, fusions, and also we provide some kind of a global whole genome view of the patients so that this can be reported to the molecular geneticist who looks all of this stuff, all of this results come to us or communicate with us, and then finally when they are ready to send the reports, they send the reports to the clinicians and doctors, and that's how basically the whole workflow works for us. So in general, what I want to end up or like summarize with that, like clinical bioinformatician, as you have seen, is we are the jack of everything and master of none. We have to be good at programming, of course. We have to learn a lot of biology because we have to communicate. We have to communicate with the also molecular geneticists and doctors. So we need to know about the biology. On top of that, we need to learn about different kind of NGS technology. So if you have one of that, then you are one of the bioinformaticians out there. This slides, this figure I got from one of the paper, and I was quite thinking about it, and in the paper as well, they said that there is a lot of skill set and a lot of people that is working together in a team of bioinformaticians. So to conclude, I probably have given you an impression that precision medicine is making strides in healthcare with a lot of different initiatives and all over the world, and especially in Sweden with the GMS, Genomic Medicine Sweden. It is making one of the applications in the common healthcare. But of course, it requires different kind of in order to precision medicine to work, it requires different kind of a computational skill as well as bioinformatics skill set. And finally, probably I have given you an idea about how a data infrastructures and how does a data bioinformatics workflow works in a real lab setting in general. Yeah, that's that. I have all about the presentation. I wanted to, end the slide as well as open the floor for the discussion. Maybe we can talk about it, like how, what are the things you should consider if you want to set up a data lab in Nepal. Most of these are my consideration, but probably there are, you guys can give their input as well. And we can talk about this in our follow-up talks. Well, yeah, that's that about it. And with that, I want to end my presentation. And if you have any questions, yeah, I open the floor for the more questions. Thank you.
Thank you for the presentation. Now, floor is open for questions if anybody's got any question.
Sailendra, एउटा त्यो स्लाइड्स खोल्नु छ, स्लाइड नम्बर 2 कि 3 हो त्यो लिंक्स वाला.
Let me extend la slide number कति
2 कि 3 होला त्यो लिंक्स वाला थियो नि त्यो
laboratory information management system. Extend la most coordinates actually. Slide number 2. अब देखिन्छ स्लाइड
माथि
स्लाइड नम्बर 2 त भन्नु होला
त्यो लिम्स लेखेको
लिम्स ल्याबोरेटरी इन्फर्मेसन म्यानेजमेन्ट सिस्टम
तल तल
यो होला
बस तल
यो
हैन यो 12 नम्बर
यो होला
ओके यो काम छ नि जस्तै पेशेन्ट आउँछ पेशेन्ट आएर डक्टरका भेट्छ त्यसपछि स्याम्पल लिन्छ त्यसपछि सिक्वेन्सिङ गर्छ सिक्वेन्सको एनालाइज गर्छ हैन अनि त्यसपछि मोलिकुलर जेनेटिक्सले रिपोर्ट तयार पार्छ त्यसपछि रिपोर्ट डक्टरलाई दिन्छ डक्टरले पेशेन्टलाई दिन्छ यो यति सबै कति दिनको काम हो खासमा कति दिनमा सबै हाउ मेनी डेज
भेरिएबल हुन्छ स्याम्पल वाइज र एसे वाइज चाहिँ भेरिएबल हुन्छ फर इक्जाम्पल जीन प्यानल छ भने जीन प्यानल लिस्ट छ भने मेबी अ विक अर सो 1 विक अथवा 2 विक टर्न अराउन्ड टाइम हुन्छ जबकि होल जिनोम सिक्वेन्सिङ गर्नु पर्यो भने अफकोर्स होल जिनोम सिक्वेन्सिङको चाहिँ एक्ज्याक्टली थाहा छैन बट 20 days अराउन्ड 20 to 30 days 25 days टर्न अराउन्ड टाइम लाग्छ होल जिनोम सिक्वेन्सिङमा त्यो बाहेक अरू ऱ्यापिड केसेस पनि हुन्छ त्यो ऱ्यापिड केसेसमा चाहिँ पेशेन्टको कन्डिसन अनुसार डक्टरलाई चाँडै रिप्लाई चाहिन सक्छ अनि त्यो चाँडै केसेसमा चाहिँ हामी चाँडै पनि एक्सपर्टिज केस अथवा ऱ्यापिड केस भनेर अर्जेन्ट केसेस भनेर हामी अर्जेन्ट केस गरेर 1 विक अन्डर 1 विक अथवा लेस देन 1 विकमा हामी रिपोर्ट सेन्ड गर्छौँ त्यो चाहिँ अब सबै जिन नभइकन अब कुनै ह्यान्डफुल अफ जिन्सहरू हैन जुन चाहिँ अर्जेन्ट केसेसमा हेरिन्छ त्यसरी पनि हामी सप्लाई गर्छौँ हामी सप्लाई भन्दा 1 विक लेटेस्टमा चाहिँ 1 विक टर्न अराउन्ड टाइम चाहिँ हुन्छ अर्जेन्ट केसेसहरुमा भनेको मतलब मैले भन्न खोजेँ के भन्दाखेरि जिन प्यानल र एसे अनुसार फरक पर्छ जिन प्यानल हो भने 1 to 2 weeks लाग्छ होल जिनोम सिक्वेन्सिङ हो भने 15 to 20 days लाग्छ बट देन अर्जेन्ट केसेस छ भने चाहिँ हामी लेस देन 1 विकमा पनि हामी डक्टरलाई चाहिँ रिपोर्ट सेन्ड गर्न सक्छौँ अनि अर्जेन्ट केसेसमा चाहिँ ह्यान्डफुल अफ जिन्सहरु मात्र हुन्छ
अनि जस्तै यो त अलिकति सबैको जेनोटाइप फरक भएकोले फरक हुन्छ अनि डक्टरले प्रेस्क्रिप्सन गर्ने बेला फेरि ट्रेडिसनल ड्रगले दिने त होला नि फेरि कि अब पर्सनलाइज ड्रग त्यस्तो त आएको छैन होला मार्केटमा
इम्युनोथेरापीहरु चाहिँ अलिअलि हुन थालेको छ अलिअलि अहिले त्यो टिएमबी ट्युमर म्युटेसनल बर्डनको अनुसार जुन अब टिएमबी भन्ने स्कोर छ त्यो स्कोरको आधारमा चाहिँ अलिअलि चाहिँ अलिअलि अहिले अलिअलि दिन त रहेछ बट देन sorry for that but then हामी चाहिँ के गर्छौँ भन्दा हामी चाहिँ मोर स्ट्रेटिफिकेसन लाइक फर इक्जाम्पल हेमाटोलोजी केसमा पनि अब माइलोइड पेशेन्ट हो कि लिम्फोइड पेशेन्ट हो कि त्यसको पनि डिफ्रेन्ट सब टाइप्सहरु हुन्छ पेशेन्ट स्ट्रेटिफाई गर्नको लागि चाहिँ हामी चाहिँ यो एनजीएस बेस्ड टुल्सहरु युज गर्छौँ लाइक भनौँ न हेमाटोलोजिकल डिफ्रेन्ट ग्रुप्सहरु हुन्छ त्यो ग्रुप्स अनुसार ड्रग्सहरु पनि डिफ्रेन्ट दिन्छ नि त त्यो ग्रुपहरु त्यो मेन पेशेन्टलाई चाहिँ त्यो ग्रुपमा चाहिँ डिभाइड गर्नको लागि ग्रुप गर्नको लागि चाहिँ एनजीएस टेस्टहरु चाहिँ गरेर हुन्छ डक्टरहरुले अहिले चाहिँ
अनि तिम्रो केही सजेसन टिप्स छ अब यति त एकदम भेग छ हैन नेपालमा गर्नको लागि त्यो पनि स्टार्ट गर्नको लागि चाहिँ कहाँबाट सुरु गर्दा हुन्छ जस्तो लाग्छ तिमीलाई
मलाई त ल्याबोरेटरी म्यानेजमेन्ट इन्फर्मेसन म्यानेजमेन्ट सिस्टममा चाहिँ स्टार्ट गर्नु भने सजिलो हुन्छ होला एउटा सानो प्रोटोटाइप वर्किङ इन्फर्मेसन म्यानेजमेन्ट सिस्टम बनाएर जसले चाहिँ डेटाबेससँग कम्युनिकेट गरेर डेटाहरु चाहिँ स्टोर गरेर र एउटा सिम्पल इन्टरफेस बनाएर भनेपछि लिम्स सिस्टम नै प्रोबब्ली मोर easy way to go around होला अहिलेलाई चाहिँ rather than एकदमै मोस्ट यसरी कम्प्लिकेटेड जानु भन्दा मोर इजी वे चाहिँ या सानोलाई इन्फर्मेसन म्यानेजमेन्ट सिस्टम बनाएर हामीले डेटालाई ट्र्याक गरेर अनि डेटालाई चाहिँ राम्रोसँग प्रिजर्भ गरेर राख्न सक्यो भने त्यति नै स्टार्ट हुन सक्छ। त्यति नै राम्रो स्टार्ट हुन सक्छ अहिलेको लागि नेपालको केसेसमा चाहिँ। यो भनेको चाहिँ अलिकति कम्प्लिकेटेड भन्दाखेरि नि ठुलो एनालिसिसहरू भएको हुनाले धेरै कम्प्युटेसन स्ट्रक्चरहरू र इन्फ्रास्ट्रक्चरहरू चाहिन्छ। हामी धेरै एनालिसिस गर्ने होइन, हामी हाम्रो डिजिटल मात्र फुटप्रिन्ट बनाउनको लागि हो भने अहिले चाहिँ एउटा सानो डेटाबेस स्टार्टअप गरेर डेटालाई स्टोर मात्र गर्न सक्यो भने राम्रै हुन्छ।
ओके, यो लिम्समा चाहिँ डेटा भनेको चाहिँ सब क्वेसनर हैन, यो त पेशेन्टसँग लिने डक्टरले।
लिम्स भनेको चाहिँ ल्याबोरेटरी इन्फर्मेसन म्यानेजमेन्ट सिस्टम हो। त्यसको नाम चाहिँ त्यसलाई चाहिँ ल्याबोरेटरी इन्फर्मेसन म्यानेजमेन्ट सिस्टमलाई चाहिँ सर्टन गरेर लिम्स बनाएको। तर लिम्स चाहिँ हाम्रो रिसर्चमा पनि लिम्स हुन्छ। लिम्स भनेको त स्याम्पल आयो, स्याम्पल कति गयो भनेर ट्र्याक गर्ने एउटा सिस्टम मात्र हो क्या। ल्याबोरेटरीमा सपोज भनौँ न कति सामान आयो कति सामान गयो टाइप्स, ब्लन्टली भन्नुपर्दाखेरि त्यसरी म्यानेज गर्ने एउटा सिस्टम मात्र हो। बुक किपिङ टाइप्स भनौँ न, हामी बुक किपिङ टाइपको एउटा सफ्टवेयर मात्र हो। त्यसलाई चाहिँ लिम्स भनेको, त्यो चाहिँ मोर जेनरल टर्म हुन्छ। लिम्स भनेको त्यो चाहिँ नेम्ड नेम होइन, एउटा त्यो मात्र हो, जेनरिक नेम मात्र हो। लिम्स भनेको चाहिँ कुनै एउटा ल्याबोरेटरीको इन्फर्मेसन सिस्टमहरू म्यानेजमेन्ट गर्ने सिस्टम हो भनेको लिम्स भनेको चाहिँ।
Thanks for the talk.
Timorko, we can talk it later, I think.
मेरो एउटा क्वेसन चाहिँ के अरे जस्तो पेसेन्टबाट आउँछ नि स्याम्पल होइन, अनि आएपछि कुन एनालाइसिस गर्ने के गर्ने भनेर चाहिँ डु यु हेभ अ सेट कि यो डिजिजलाई यो गर्ने अनि त्यसको आउटपुट दिएपछि अनि मोलिकुलर जेनेसिसले डिसाइड गर्छ कि भन्नाले हु डिसाइड्स स्याम्पल त डक्टरले दिन्छ अनि यु डु अल द सिक्वेन्सिङ टुल्सहरू युज गर्ने भयो होइन। तर त्यो कहाँ कुन पेसेन्टमा के युज गर्ने भने चाहिँ इज इट अ सेट कि कस्तो खालको हुन्छ भन्ने?
सेट भन्दा पनि डक्टरले नै, अब पेसेन्ट आउँछ डक्टरलाई चाहिँ शङ्का लाग्यो हुन्छ, उसले चाहिँ ओके मेबी यो पेसेन्टमा चाहिँ यस्तो यस्तो फेनोटाइपहरू देखेको छ, प्रोबेबली चाहिँ यो टेस्ट गर्दाखेरि सही हुन्छ भनेर डक्टरले अर्डर गर्छ युजली। अनि डक्टरले अर्डर गर्नुको साथसाथै के हुन्छ भने अरू अर्थोगोनल मेथड्स पनि हुन्छ यो बाहेक, लाइक फर इक्जाम्पल बायोप्सीहरू गरेर इम्युनिज स्टेनिङहरू गरेर पहिला प्रिलिमिनरी इन्फर्मेसनहरू पनि लिएर ओके यो पेसेन्ट चाहिँ सायद हेमोलोइड भनौँ न क्यान्सर कुनै शङ्का लागेको छ भने डक्टरले पठाउँछ। अनि डक्टरले पठाएको रिपोर्ट डेटा पठाएको इन्फर्मेसन अनुसार हामी चाहिँ त्यसरी प्रोसेस गर्छौँ। बट कतिपय केसेसमा चाहिँ हामीले कम्प्युटेसनली पनि डिटरमाइन गर्नुपर्ने हुन्छ स्याम्पल राइट छ कि रङ छ भनेर। जस्तै स्याम्पल मिक्सअप हुनसक्छ, फिमेललाई मेल मेललाई फिमेल हुनसक्छ, त्यो सबै कुराहरू चाहिँ हामी कम्प्युटेसनली पनि चेक गर्छौँ। बट बेसिकली तपाईँको क्वेसन एन्सर गर्नु पर्दाखेरि चाहिँ इट्स डक्टर हु डिसाइड्स इट, बट अन टप अफ द्याट वी हेभ अदर मेथड्स एज वेल टु डिटरमाइन विच सुड गो देयर भनेर स्टाफले पनि।
थ्याङ्क यू। Do we have any more questions? अनि ल्याङ्ग्वेजमा जावा र पर्ल अघि त्यहाँ प्रेजेन्टेसनमा तपाईँले भन्नुभएको छ पर्ल युज गर्छ भनेर होइन, अनि के डिफरेन्स हुन्छ कि इज इट काइन्ड अफ सेम भन्नाले त्यसको काम नै डिफरेन्ट हुन्छ कि?
काम सबैको एउटै हुन्छ, पाइथनहरू सबै काम चाहिँ एउटै हुन्छ, तर लेख्ने स्टाइलहरू चाइनिज ल्याङ्ग्वेज जापानिज ल्याङ्ग्वेज जस्तै हो, डिफ्रेन्ट सिन्ट्याक्सहरू हुन्छ, डिफ्रेन्ट कोडिङ स्टाइल्सहरू हुन्छ, त्योहरू चाहिँ डिफ्रेन्ट हुन्छ। पर्ल चाहिँ युजली लेगेसी भन्दाखेरि पहिलादेखि नै चलिआएको हुनाले चाहिँ यहाँ पनि धेरै जसो पहिला पर्लमै एकदमै युज गर्नुभएको हुनाले चाहिँ पर्ल युज गर्छ हामी चाहिँ अब मोर न्यू जेनेरेसनहरु चाहिँ मोर पाइथन युज गर्छौं बट क्वेशन भन्नुपर्दा इट्स डज द सेम स्टफ बट सिन्ट्याक्स चाहिँ डिफरेन्ट हुन्छ सिन्ट्याक्स डिफरेन्ट र इजी र अलि डिफिकल्ट टु लर्न काइन्ड मात्र डिफरेन्ट हुन्छ बट सबैको काम एउटै नै हो
थ्यांक यू So do we have any more questions? No, I think looks like questions about China. It's been a pleasure being with you all today. Thank you very much, Dr. Sailendra Pradhananga, for delivering your talk. And we hope that today's talk has been informative. And thank you very much again. And yes, with this, I'd like to close the session. Thank you.
Thank you very much. Have a wonderful day ahead.
Thank you.